So, respiration, already I said definition, what happens, the breaking down of the food material in the presence of oxygen is called respiration. So, respiration is two types. So, here they given that anaerobic respiration and fermentation, aerobic respiration. So, the major differences between aerobic and anaerobic. So, first of all, let us go to the similarities. Similarities. So, in aerobic, anaerobic, anaerobic respiration. Let us in aerobic respiration, to carry out aerobic respiration, oxygen is required. Oxygen is required. To break down, glucose is required, and which CO2 is released, and energy is released. So these are the in respiration. First point is what, to carry out aerobic respiration, oxygen is required. Then food material, glucose is required. Then carbon dioxide is released, energy is released. Now anaerobic respiration. So there is no oxygen. There is no oxygen, but breaking down of the food material, glucose is takes place, and releasing of carbon dioxide, energy. So the similarities is carbon dioxide is releasing, energy is releasing. So these are the similarities. But differences means anaerobic respiration does not require the oxygen. Aerobic respiration have the require the oxygen. So now you can see that the another difference is the CO2 which are released in the aerobic respiration are six CO2s, six CO2s. But whereas in anaerobic respiration, the CO2s are two CO2s only. So the major differences between this one only. So in aerobic respiration, carbon dioxide molecules released six CO2 molecules, whereas in anaerobic respiration, two molecules of CO2s. So, and also energy is released in stepwise manner, but here energy is released at once as a heat. So let us go to that. So which material is break down in the cell? Glucose. So after breaking down the glucose in the respiration, pyruvic acid is formed. For one glucose molecule, two pyruvic acid molecules are formed. These two, while converting glucose into pyruvic, energy is released. That process is called glycolysis. Glycolysis is nothing but breakdown of the glucose molecule is called glycolysis. Breakdown of the glucose molecules is called glycolysis. So this glycolysis, it takes place in both conditions: presence of oxygen, absence of oxygen. So it is common. Now this pyruvic acid is to enter into aerobic and anaerobic, oxygen is determined. If the pyruvic acid is find, if the pyruvic acid is find the oxygen, and then it enter into the, if the oxygen is available, if the oxygen is available, the pyruvic acid is enter into the aerobic respiration. If the oxygen is not available, the pyruvic acid is enter into the anaerobic respiration. So the way entering aerobic and anaerobic of the pyruvic acid is determine the presence and absence of oxygen. If the oxygen is present, pyruvic acid is enter into the aerobic. If the oxygen is present, absent, it is enter into the anaerobic respiration. So what is the main result during anaerobic respiration? During anaerobic respiration, glucose is converted into lactic acid. Glucose is converted into lactic acid in the muscles of human body and animals, and energy also released, somewhat energy. So lactic, so the energy is how much means 56 kilocalories. Low amount of energy is released, 56 kilocalories energy is released, that energy percentage. And which bacteria is converted glucose into lactic acid means lactobacillus bacteria. Whereas another important one here, microbe, especially that yeast belongs to fungi. Yeast, it belongs to fungi. When due to absence of oxygen, The glucose is converted into ethanol, and that is ethyl alcohol. Ethanol is also called ethyl alcohol. So 2C2H5OH, the chemical formula of ethanol is 2C2H5OH. 2C2H5OH, ethanol. So how much carbon dioxide molecules are released? Two, two molecules, two CO2. And energy I said already, 56 kilocalories energy is released. So this is the anaerobic respiration chemical formula we can say. So ethanol is the formula is 2C2H5OH, two molecules of CO2 and 56 kilocalories energy. So like this we should write the chemical equation. The end products of anaerobic respiration, three: ethanol, nothing but ethyl alcohol, carbon dioxide, energy. Ethanol or ethyl alcohol chemical formula is 2C2H5OH. So how many molecules of CO2 are released? Two CO2s. And energy how much? 56 kilocalories. So this process is called fermentation. So it is important in examination level. Fermentation, which means the process of conversion of glucose into ethanol or ethyl alcohol by the yeast. The process of conversion of glucose into ethanol or ethyl alcohol in the absence of oxygen by the yeast is called fermentation. By yeast is called fermentation. So fermentation definition once again, the process of conversion of glucose into ethanol or ethyl alcohol by the yeast in the presence of in the absence of oxygen is called fermentation. Fermentation is one type of anaerobic respiration. So anaerobic respiration chemical equation, in sometimes what happen, glucose is converted into lactic acid. So, and energy. So the chemical formula of ethanol is 2C2H5OH. So how many CO2s are released? Two CO2s. How much energy? 56 kilocalories. This is the anaerobic respiration. When you come to the aerobic respiration, when the presence of oxygen, pyruvic acid is entered and converted into carbon dioxide. How many carbon dioxide molecules? Six CO2s. How many molecules of water? Six H2O. How much energy? 686 kilocalories energy is released. 686 kilocalories energy. So when we compare with the anaerobic, here 56, how many carbon dioxide? Here 6 CO2s and here 2 2 CO2s. So how many water molecules? 6 H2Os. So that is the main differences. So releasing of carbon dioxide energy is common anaerobic and aerobic, but percentage is different. Whereas in aerobic, 6 molecules of water and 686 kilocalories of energy is released, and six carbon dioxide molecules are released. That is, you have to keep in your mind in anaerobic respiration. So here, this experiment is during strenuous exercise. That is, fastest sprinters do not breathe while running. They won't breathe due to at the time what happened, oxygen is decreases. So oxygen is decreases at that time due to absence of oxygen. The glucose which is present in our body muscles convert into lactic acid. So if the lactic acid is remain in our body, it get body pains. That is, you have to keep in mind. So this is the experiment. This experiment. So due to loss of while running, so oxygen debt which has to be repaid later. So during running, what happened? oxygen percentage decreased. When we relaxed, then it will be again will regain the lost oxygen. So if at the time what happened, due to lack of oxygen, the glucose is converted into lactic acid, and then this lactic acid is slowly converted into as a carbon dioxide, energy, and water when the oxygen is present. So the presence of lactic acid in the blood is the main cause of muscle fatigue. It is important in examination level. Choose the correct answers, it will come. If the lactic acid is remain in our body muscles, what happen? Body muscles get pain and also fatigue. So and tiredness. When it is regain the oxygen percentage, again we will lose the body pains. So that is your keep in your mind. Now you go to the anaerobic respiration. So anaerobic respiration definition, if the respiration is carried out in the presence of oxygen is called anaerobic respiration. So this is experiment with yeast. It is proved by, so this experiment is the most important here uh, to prove the anaerobic respiration, to prove the anaerobic respiration. This experiment with yeast అనగానే మనకు anaerobic respiration గుర్తుకు రావాలి. Anaerobic respiration definition what? It takes place in the absence of oxygen. So when living organisms kept away from supply of oxygen, yeast grows rapidly if it is supplied with the glucose in the solution. So it need, so now let us go to the apparatus. So during anaerobic respiration, production of heat and carbon dioxide during anaerobic respiration. So that we need a test tube and thermos flask, thermometer, liquid paraffin, and lime water. So here, first of all, we have to take the glucose solution and pour it into the thermos flask. So this glucose solution is must be boiled and cooled one, boiled and cooled one. So after pouring the glucose solution, you have to leave the yeast cells. You have to leave the yeast cells. So now on that you have to pour the liquid paraffin on the surface of the glucose solution. Why? Because this liquid paraffin not allowed the oxygen to reach to this glucose solution. So here which experiment we are doing anaerobic, so that we we have to avoid the attachment of the oxygen to this solution. To avoid oxygen attachment of this, we have to pour the liquid paraffin. This paraffin it prevent the not to enter oxygen to this glucose solution. Now kept midst of thermos flask a thermometer into the glucose solution, and another attach a tube from this thermometer a thermoflask to the another test tube which containing the lime water. Now this entire what's happening? Yeast undergo anaerobic respiration. During anaerobic respiration, heat is liberated. Then it in thermometer mercury level is increases. Whereas during anaerobic respiration, yeast produce carbon dioxide. It leaves and enter into these tubes, and finally this carbon dioxide moves to into the lime water test tube. This carbon dioxide is convert the lime water into white precipitation. White precipitation. So during anaerobic respiration, carbon dioxide is releasing. We can prove this one. Heat is liberating. How can we prove? Because of expansion of mercury level in the thermometer. So both we can see. So here these three points most important in examination level. So how can we remove dissolved oxygen from glucose solution by heating it for a minute, then cooling it without shaking? Now put in some yeast. The supply of oxygen from the air can be cut off by pouring 1 centimeter layer of liquid paraffin onto the mixture. So this, first of all, to remove the oxygen in the glucose solution, we have to heat. Then after that, cool and pour the this glucose solution into thermal flask. And not to attach the or contact the oxygen to this heat and cooled glucose solution, we have to pour the liquid paraffin on this glucose mixture. Now, then if you wish to check that oxygen is, it is important point, most important in examination level. Second point, even if you want to test oxygen either removed from the mixture and add a few drops of diagen green, Janus green B solution to the yeast suspension before you pour the liquid paraffin over it. So before pouring the liquid paraffin, you have to add the few drops of diagen green B, that is Janus green B indicator solution. So when the oxygen is not present in that solution, now the blue turns into pink. Blue, the blue diagen green B turns into pink. So it indicates that there is no oxygen in the glucose solution. Then you should carry out the experiment and you pour the liquid paraffin onto the boiled and cooled glucose solution which is present in the thermos flask. So another bottle containing bicarbonate or that is indicator solution, nothing but lime water is attached to during this process. Anaerobic respiration amount carbon dioxide is released and that should be converted into white lime water. Already I explained. So the carbon dioxide passes through the tube and turns lime water white milk, water milky. We observe the changes in the temperature also during this process. So carbon dioxide production part of the experiment on the smaller scale using test tube. If you so warm them to about 37 degrees centigrade in order to speed up the test. What happens when a baker prepares a dough by mixing yeast in it? What happens? It makes swell. Yeast Saccharomyces cerevisiae fungi, which helps to the make this makes the dough into fluffy nature, smooth and soft nature due to absence of oxygen. So now already I said next it is important. I said fermentation. So once again listen, fermentation definition: the process of conversion of glucose into ethyl alcohol by the yeast. By the yeast is called fermentation. The result of fermentation, if it is yeast, it is participated. The result is formation of ethyl alcohol or ethanol and carbon dioxide, two CO2s, and 56 kilocalories energy is released. So this anaerobic respiration is mainly used in brewing industry to preparation of beer, whiskey and rum, brand, etc. This fermentation is used in brewing industry. So here they given the ethanol can be separated from the yeast glucose mixture by the process of fractional distillation. By the fractional distillation, ethanol is separated and ethanol boils at lower temperature. Then the sugar solution collect information about fractional distillation. You know that fractional distillation by this process we will get different products by heating different centigrades. సో ఏదైనా ఒక సొల్యూషన్ని మనం డిఫరెంట్ సెంటిగ్రేడ్స్కి హీట్ చేయడం వలన మనకు డిఫరెంట్ స్టేజెస్లో డిఫరెంట్ మెటీరియల్స్ రావడం అనేది జరుగుతుంది.